Well, all right, folks. We're at 6:30 here this evening. On behalf of the Soil Health Academy board members David Brandt, Kim Barman, and Don Brightcruz, alongside with the Partners Understanding Ag, we welcome you to tonight's webinar with Dr. James White. Dr. White is a professor at Rutgers University. He's the author of more than 200 articles, an author and editor of reference books on the biology of endophytes. Before we get started with Dr. White this evening, we do want to let you know we have future webinars coming up next week, April the 7th at 7:30, Tips for Successful Grazing Season with Dr. Dr. Alan Williams, I apologize. On April the 9th, Marketing, the Tables Have Turned with Dr. Alan Williams. April the 14th, Marketing webinar, Laying the Foundation with Gabe Brown, Dr. Alan Williams, and myself, Shane New. And April the 16th, Soil Building Secrets with Ray Archuleta and David Kleinschmidt. Dr. White, I've got to ask you this question. What got you down this path studying about endophytes as long as you have?
Well, when I was an undergraduate student at Auburn University in Alabama, I actually was working with a faculty member there who was working on this fungal endophyte in forage grasses. And as an undergraduate, I got to study in the fungal endophyte. And it was really interesting. Then when I went to University of Texas to work on my PhD, I decided to study these fungal endophytes in grasses much more. And so I did my PhD studying those fungal endophytes. And the thing that excited me about it, and still excites me about studying microbial endophytes, is that it's a mystery. You see a plant, you don't see any microbes in it, you just see it as a plant. But if you study it very closely, if you use microscopic techniques to take a look at it into the cells and in between the cells, you spend enough time, you begin to see that there are actually microorganisms that are inhabiting plants. And so it turns out that a lot of plants, all plants really have these microbes in them, and the microbes have some really interesting effects in the plant.
And that's what you're going to be discussing with us tonight in your PowerPoint, the rhizophagy cycle. So we will let you get started, and we're all anticipating your presentation.
Thank you, Shane. So what I'm going to try to convince you of by just showing the research that we've done, I'm going to try to convince you that plants are actually cultivating microbes in the soil microbes around their roots, and then they're internalizing those microbes and they're extracting nutrients from those microbes, and they're becoming, essentially, those microbes become endophytes. And when they're inside an endophytic phase, the plant extracts nutrients from them. And then the plant will eject those microbes, extracted of their nutrients, free of the nutrients or with minimal nutrients. They'll eject them back to the soil, and then those microbes will go and get more nutrients and so forth. So the plants are actually cultivating these microbes, and I'll show you the details of how that's working. Something's not working right, it's not forwarding. Okay, can I stop share now?
Yeah, I think so, Kathy.
Gonna have to do something to see what's going on.
Hold on a second.
You can go ahead if you want, and then we can
Okay, I'm going to stop it. I'm going to see what's going on. Okay, I'm going to go back to share again. And
Okay, we see your screen now, your first slide.
okay,
There we go.
it worked.
Yes, sir.
Sorry about that. Okay, so these microbes are endophytic. And what I'm showing you on this screen is actually an endophyte. And this blue line here, you'll see my arrow. This is an endophytic fungus in a grass. And in the background, you see the cells. I'm showing you the outlines of the cells there, of the parenchyma cells. And this is from a tall fescue plant. An endophyte is any microbe that goes into a plant and doesn't cause disease, doesn't have any outward signs of its presence, and it basically has effects. Usually they're positive effects on plants. Turns out endophytes are everywhere. All plants carry endophytes, and those endophytes are in seeds. Their endophytes are recruited from the soil. They come out of the soil and the plants take them in. Every plant actually has a community, a small community, or even you might consider it a large community in some cases, of endophytes. These are hemp seeds, and hemp seeds, a lot of seeds will carry endophytes with them. Hemp is no different. With hemp, if you germinate the hemp seed, you get that little root that comes out, and you can see these little structures here. You see the arrows are indicating the root hairs. Those are root hairs. And you can see all those little brown structures inside those root hairs. Those are the bacteria. You see the image to the right over here. See those little spherical structures. Those are the bacteria inside that root hair. And those bacteria, actually in this phase, they don't have cell walls there. They are spherical structures. They're spherical protoplasts, and they're just actually outside the membrane of the plant and of the root cell there, that hair cell. So you can kind of see them sticking on the outside of that. And you can see the structure out here, that's actually the cell wall. So there's a gap. In this case, you can see where my arrow is. There's a gap between the cell wall and the membrane and the layer of microbes on that membrane. This is another hemp root hair. And you can see here at the tip, you see the brown. That's where the bacteria are. And you can see the blue there. That is the bacterial cytoplasm right there. You can see those bacteria. Some of those are actually rods there. These bacteria essentially are fished out of the soil by plants. And what they do is they will take these bacteria out of the soil, the ones that are most useful for them, they will then deposit on their seeds. So that seed, when that seed falls somewhere and it starts to germinate, it'll have a little community of microbes that go with it. This is the natural way that seeds work. You do anything to clean those seeds, remove the microbes on them, or you grow seeds in substrates that don't have microbes in them, you're not going to have a rich community of microbes on those seeds that are produced. This is some cacti on a desert island of Bonaire. And this is one of the good things about my work is I get to travel around to study plants in different places. And so these are some desert plants. At the top of these plants, there's some fruits up there, way up there. And you get those little fruits off. You get the seeds out. And there's the cactus seeds. Germinate those seeds, and you have a little seedling here. can see the roots down here, kind of orange color. You can see the root hairs all over them. Here's the shoot of the cactus. And if you look at that microscopically, these are now the root hairs. These root hairs are filled with microbes. All these little dark structures there, those are all the bacteria in those hairs. And you can see here, now this is a root hair, and you see the little clusters of bacteria there, the spherical clusters. You can see in some cases, they're like in pairs, like right in there, you see pair, you see a pair there. Those are recently divided, those in the microbes. Now, actually, these microbes are not in the root cell itself, but instead they're on the surface of the root cell plasma membrane. So they're stuck out there on that plasma membrane, but they're underneath the cell wall there for that of that hair. These endophytes have some profound effects on plants, and among those effects are these. First one, they improve plant stress tolerance, and what happens is they go inside the roots, inside the plant cells, and they elicit the plant to secrete reactive oxygen onto them. And when the plant does that, it gets nutrients from the microbes, but it also causes the plant to upregulate antioxidants and other kind of oxidative stress tolerance genes. And that makes the plant more tolerant to all kinds of oxidative stresses, which could be high salt, could be high temperature, could be low temperature, whatever it is. It could be metals, heavy metals or something in the soil, whatever it is that causes oxidative stress. So the endophyte makes a plant stronger and more stress tolerant. The endophytes, some of these endophytes will actually go outside the plant into the soil, and then they will colonize pathogenic fungi. And they will cause those fungi to actually leak nutrients. And those fungi will leak nutrients, and it will cause those fungi to become less virulent. Because they don't have, because they're leaking nutrients, they won't have the oomph that it takes to actually cause disease. The other thing that these endophytes do is they will modulate root development. And that is when a microbe is on seeds and a seedling germinates, if the microbe is there, what happens is the seedling root will grow down into the soil and will also form root hairs. But if you remove the microbe, what happens to the root is, if we say sterilize the seeds, take all the microbes off, what happens is the root will stay on the surface of the soil, or in some cases it will actually grow into the air, and no root hairs form. So those microbes have to be there for this gravitropic response, for the roots to grow down, and for root hairs to form on seedling roots. So it's very important for that development of the plant. Also, because these microbes function to liberate nutrients in the soil and also carry some of those nutrients back to the plant, the presence of the microbe improves nutrient absorption into the plant. It's actually very important for that nutrient absorption function. And finally, the endophytes, the endophytic microbes in plants, they will, or certain ones at least, will cause plants to develop a different chemical, different chemicals internally. And I'll show you an experiment where we used microbes to increase the production of carotenoids or carotene in carrots. With the microbes, you'll see the carotene. Without, you'll see less carotene. Okay, so about 10 years ago, some Australian scientists discovered that plants were internalizing yeasts and bacteria. And what they did actually is they labeled the microbes with a fluorescent stain, green fluorescent protein tag, essentially. And then they fed it to tomato plants and some Arabidopsis plants and then they tracked it inside the plant. And you can see these images here where actually there is a root hair here with my little arrow, and you can see the microbes. Those are yeasts glowing from that stain that they put in there. And you can see over here, you can see the root cells there, and you can see the microbes, the glowing microbes inside those roots here as well. Here as well, you see those microbes in there. These investigators saw that after a period of days, the fluorescence stopped. And so it appeared that these microbes were being degraded inside the roots. And because of that, they envisioned this process as essentially a microbial eating by plants, by plant roots. And they called it rhizophagy, rhizo for root, and then phagy meaning eating. So they called this process rhizophagy. And what we have actually learned over these past few years is that it's actually more of a cyclic process. And that is what's happening is that plants are internalizing microbes. They're attracting microbes to the meristem, to this growing point, the growing root tip here. And then they're somehow internalizing those microbes into those cells. They take them into the cells, into the root cells, and then they will subject them to a form of reactive oxygen called superoxide. And that superoxide strips the cell walls off of the microbes and then extracts nutrients from them. And this is an active process. I'll show you how that works. But eventually, what happens is the microbes that survive that process will trigger root hairs. And those root hairs will start to elongate. And as those root hairs elongate, those microbes will be ejected back out into the soil, where they'll go back into the soil, they'll acquire more nutrients, and then they're attracted back to the root tip again. And what's attracting them to the root tip actually are exudates. And this is at this root tip, this is where the plant will produce exudates. And so it actually is, the plant is secreting some exudates out that tip, and it makes a kind of an exudate trail in which the microbes can follow and then get back to the plant where they're internalized again. Okay, so this is a grass seed, and it's germinating grass seed. And you can actually see here right around the root, you can see there's some yellow there. Those are the microbes that have come off of that seed and are colonizing that root. And actually here at the shoot, you see some yellow also. Those are microbes there as well. Some microbes are colonizing also the shoot tissues. Well, it turns out with grasses in particular, on the endophytes, the microbes are actually carried on the surface on the tissues, the dried plant tissues that adhere to that seed surface. And so that's kind of that's the vector for those microbes. And it turns out there's 3 or 4 or 5 different microbes. It's a small community that get carried on those seeds. This is a diagram of a root, and the root tip down here, and you can see the hairs forming back here, and you see this exudate zone here. Okay, this is the zone where the plant is secreting carbohydrates, and in some cases some amino acids and some organic acids and so forth, essentially to feed those microbes, to cultivate those microbes, and to attract those microbes to that zone so that the plant can then increase them and then internalize them. Okay, so this shows a Phragmites root. This is a grass. This is this big common reed grass that is a nuisance to a lot of people. It's invasive Phragmites. But you can see the root tip here. All this blue. These are where the cells are soft, have soft cell walls. They haven't hardened here. And you can see this blue zone out here. Those are the microbes. Those are all those little dots out there are microbes that this plant root is cultivating right there at the tip. And it is underneath this zone where it's blue, it is internalizing, taking in those microbes into the cells, and right there in that zone. And this actually shows a grass root, corn root, and you can see these corn roots have lots of little branch roots there. All those branch roots have little tips on them, and those little tips are where those microbes are being cultivated and internalized. So plants that have more of these kind of root tips, have more branching, like grasses that have more branching roots with lots of little root tips, they're doing this rhizophagy cycle a lot. They're taking in all those microbes at all these little tips. So they're very heavily relying on these microbes. What happens once the microbe is internalized, the plant cell, the root cell, will actually secrete superoxide. You can see here the red superoxide. This is the compound. It's basically just an oxygen that's highly, highly reactive, and it's highly potent. And what it does is it will zap those bacteria, and it'll zap the bacteria and essentially oxidize off the cell walls off of those bacteria, so that they become naked protoplasts. And okay, so this is basically shows that process. Here to the left, you can see all these rods. Okay, these are all the bacteria that have their cell walls. They're rod shaped. After the superoxide hits them, you can see here, they become these little spherical protoplast structures. And these things, these spherical structures, they lack their cell walls, and they also lack their shapes. They don't have rod shapes. And the other feature of these is they're many different sizes. There's little ones and there's big ones, and then they bud. They bud very fast. Actually, when these are inside in a living root, what we see is that the plant actually is moving these around, circulating these and breaking these up. And so it essentially is replicating these microbes, essentially cloning these microbes. And then by the time that the root hair is fully developed, there'll be lots of these clones of these microbes that carry nutrients for the plant. And then it squirts those cloned microbes, the good ones, back out into the soil. And so populates the soil with those good microbes. Okay. The plant takes the cell walls off the microbes, off the bacteria. And why is that important? That, for one reason, that's important. Because those cell walls carry nutrients, and the bacterial cell walls have what we call peptidoglycan, which is a protein substance. And so that those peptidoglycans get stripped off of the bacteria, and then the plant can absorb those as a source of nitrogen. So it's a source of nitrogen for the plant. Okay, this is a fescue root, seedling root, and you can see the root here, and you see all these root hairs here. And this is actually stained for superoxide, and the superoxide we use a stain called NBT, but it stains it blue wherever the superoxide. And you can see where those out here, these little at the tips of the hairs where all that blue is. Those actually where the bacteria are accumulating in those hairs out at the tips. And so that's where the superoxide is really zapping them. But of course, when they're moving around, they're also being hit with superoxide. I'll show you that. Okay, so this shows one of these grass root hairs. And you can see in here the little bubbles all in there. And you can see the blue, it's stained for superoxide, the purple there. And you can see there's some of them are really dark from that. And you see some here at the tip that aren't as dark because superoxide actually is short-lived. So unless it's touching the membrane of the root cell, it's not going to be purple. It won't get exposed to the superoxide. So many of them accumulate out here at this tip of the hair, and they actually are ejected from the tip. But you can see they're spherical structures. So, and these are just loaded. These actually are fat with microbes. When we remove all the microbes, they're not fat hairs like that. They're little skinny, almost needle-like structures. The protoplast itself, the root hair itself, is really skinny there at the tip. So it's fat because there's so many microbes out there between the cell wall and the protoplast itself. So these hairs just get fat with microbes. Here's another one. You can see all the superoxide there. You can see the microbes out there. They're again outside the plasma membrane and but underneath the cell wall. One of the techniques we use to look at these microbes is something we call reactive oxygen staining. And basically we use this substance we call it DAB, but it has this long name, diamino tetrahydrochloride. You can see why we just call it DAB. But what this does is it will actually stain for hydrogen peroxide. And so on this slide, you can see the seedlings, and you can see the brown around there. The brown is where hydrogen peroxide came out of those roots. And so what happens is when these microbes are inside the cell, they're hit with superoxide, but that superoxide breaks down and you get the formation of hydrogen peroxide. And so that's the brown that we can see using this staining technique. Okay, this is one of the Phragmites, that big grass root cell here, root epidermal cell. And you can see these little brown things. These actually show the bacteria, these bacterial protoplasts inside the Phragmites cells. And you can see they're actually, you can see this arrow here. There's like 3 in a line here. You can see the big one, that's the oldest one. And you can see the smallest one there, that's the youngest one. As it turns out, what happens, some of these, as they age, will get hit with superoxide, and then they'll lose their protein. And so they lose this blue staining. This blue is from a protein stain we call aniline blue. And so the little ones still have all their protein in them, and they show that blue color. Okay, so the older ones will actually get fully degraded inside. So there is a degradation that happens to some of these cells inside. So microbes, if they're inside, will trigger the gravitropic response. They'll affect development. And they will also trigger root hair elongation. But they'll have another effect. They'll cause increased root branching, and they'll cause increased root and shoot elongation. Okay, so they affect development. This actually is the root of a grass called Bermuda grass. And you can see the root tip over here, and this is stained for the DAB. We're using the DAB for hydrogen peroxide. And this actually to the right shows the older part of that seedling root. You see there's no hairs there. What we did is we removed all the microbes from the seed coat in this case. And in order to get rid of all the microbes, you actually have to sterilize a lot. A lot of people will sterilize 5 or 10 minutes and think that they've done a good job sterilizing, and then they'll go do their experiments. But what we do is we have to sterilize for about an hour using like 3% sodium hydroxide, not sodium hydroxide, but sodium hypochlorite, bleach essentially, 50% bleach for an hour with agitation, and to remove all those microbes. And here is when we put the microbes back on. Here what we did was we took one of our Phragmites microbes, a Pseudomonas, we put it back on the root, on the seed essentially, not the root, but the seed of the Bermuda grass. And then we germinate it, and you see hairs forming immediately after the root tip. Hairs are forming. So the microbe is stimulating hair development, but also what you can't see is that these hairs also grew down into the agar, we did this experiments on agar rows, so they grow down into the medium here with the microbes. This actually is a root that's been squashed, stained for hydrogen peroxide using that DAB stain, and this is right around that meristem. And so you can see here, I'm circling these meristematic cells, little square cells here. You can see here's a big one there. These are still dividing actually, but you can see this brown structure here, and you can see it's rod shaped. That's that pseudomonad, that's that bacterium inside these. You can see all here, they're all out in here. All of those pseudomonads, or many of those pseudomonads were internalized. And so when they're internalized, they still, immediately afterward, they still have their cell walls, and so they still have their rod shapes. It's only later, once the superoxide hits them, that they lose that shape. Okay, so this actually shows a root a little bit more developed, away from the root tip. You can see these hairs, and see all the brown things in there. All those are the bacteria. All those brown dots are the bacteria that are inside those root hairs. And you can see, if you look at the root self, the root body itself, you can see little brown dots in there. Those are all the bacteria inside those root cells. This is a close-up of a hair, and you can see the spherical structures in there. Those are all those pseudomonads inside that root hair. These root hairs become filled with these bacteria. They just become fat with these microbes. Tomato actually will carry in its seed, in its fruit, and on its seed, and we think maybe in some cases, maybe also inside the embryo itself, because it's very difficult to clean all the microbes out of tomato. But we have some endophytes in there. And the endophytes, this is from a seedling. This is actually hairs from a tomato seedling that germinated. So this is on the root of a tomato seedling. And you can see these little packets of 4 cells here. 1, 2, 3, 4, looks like they're a little cluster of cells. So we isolated this endophyte out of these tomato plants. And as it turns out, it's a bacterium called Micrococcus luteus, and it forms its cells forms these little clusters of 4. So we could track it, we could see it, we could see when it goes in, because we'll see the tetrads when they go in, and then when they turn to the protoplasts, when they lose their cell walls, we can see that too, because they'll become spherical structures. And what we tend to do is we'll take microbes out of one plant, and then we'll put them in another plant, another test plant, to see if it does the same thing, to see if it goes in. And so that's what we did in one set of experiments, where we used curly dock, Rumex crispus, which is a common weed around farms and roadsides and everywhere, basically everywhere. But we sterilized those seeds, and then we put our micrococcus on it. And right here to the left, you can see right around the meristem, you can see these little tetrads. See the arrows here, little blue tetrads. Those are the micrococcus where they went in. That's right around the meristem. This one to the right is around the meristem in an area on a root or a seed that we did not put this micrococcus back on. And so we see no tetrads here. That was our essentially non-inoculated control. This is a close-up of that, just so you can see the tetrads inside. They're inside the cells, right around that meristem zone. This is what happens after the superoxide gets ejected or put onto the microbes. You can see they're spherical now, and they no longer form tetrads, and they're forming these chains. And you can also see that these are actually, in some cases, it looks like the membrane goes down. You can see these are actually on the membrane, or at least appear to be on the membrane itself. So that membrane is pulled away from the cell wall a little bit. Here, you can see that a little bit better. You see the membrane folding down, and you see those microbes on that membrane there. We always see it like this. We very rarely see these that go into the cytoplasm. We think some do, but we're not certain of that, and we can't really confirm that that's happening yet. But most of these stay right outside the cytoplasm. Here is a root hair, tomato root hair, in this case, or I think this is actually that curly dock, Rumex, and you see the protoplasts inside those hairs. Okay, this shows actually in some cases we can see the bacteria, those protoplasts, turning back to the walled shape while they're inside the cells. Okay, so this to the left shows those bacteria in culture. So we grow them on agarose with lots of nutrients, and they look like that. You see the tetrads there, clusters there. And here is actually inside one of the cells. And I think this is a dandelion. Actually, that's one of our test seedlings, dandelion, another weed. And you can see these little structures out here. These are actually the protoplasts. And here's one that's going back to tetrads. And you can see the 1, 2, 3, 4 parts. Here also, these are all these little things here. Those are all protoplasts. And you can see them going back to the tetrads now inside the dandelion cells, root cells. So what happens is the root hairs will actually eject those microbes out of the tips of the hairs as those hairs elongate. And so this to the left here, you can see a root hair on that Rumex crispus again, or that curly dot. And you can see the tip there. You see all these little tetrads actually forming at the surface. And you can almost see little channels through that wall. That's what happens is little pores form. And those protoplasts are forced, or I like to say squirt, out of those pores. And so they accumulate outside then. And then what they will do is they're very tiny. They're without their cell walls. They'll immediately reform their cell walls and they'll grow. And then you can see the younger ones here and the older ones are spilling off here. These are bigger bacterial cells. This is a close-up of one of those tips. And you can see here, this is a tip of a root hair. And you can see these little spherical structures. Those are the protoplasts underneath. And you can see the channels right there, kind of greenish channels, where there's a pore and where those microbes are being pushed out. And then they're reforming the cell walls here. And for microbes that have flagellae, they'll reform their cell walls and they'll reform their flagella, and then they'll swim out into the soil. And so they're actually being moved by the plant back out into the soil to acquire more nutrients. And presumably the ones that go all the way through the rhizophagy cycle and come out, those are the best microbes. Those are the microbes that carry the nutrients for the plant. Those are the ones that the plant then essentially is repopulating the rhizosphere in the soil with those helpful microbes. So this actually is another one of these young root hairs, and you can see the little bacteria here reforming their tetrads on the surface, and they're small, and you see them spilling off, continuous layer, and getting bigger as they do, as they spill out their microbes there as they mature. So what is the function of the root hair? You know, we all know from 3rd grade or 2nd grade science that root hairs function to absorb nutrients for plants, and that's how they work. And they increase the surface area and they suck in more nutrients. Well, I mean, that may be, but in terms of the rhizophagy cycle, they have this other function. And this other function is that the root hair actually is the mechanism that, or like elevators, in which the microbes are moved from inside the plant back out into the rhizosphere, and they can be deposited in the soil rather than being released right on the surface of the root where they can't acquire any more nutrients. So the plant actually moves them out there into the soil and then ejects them. So to the left here, this is an image of a root, and you can see the tip here. You can see the microbes around that tip. That's where the plant is cultivating those microbes, around that tip. And you can see this, they're internalized, and then the microbes trigger hair development, and then they're ejected out here away from that root. Okay, this actually shows, this middle one shows some root hairs. You can see the microbes. This is a fluorescent stain. All these little green fluorescent things, those are all bacteria that are being ejected out of those root hairs. The one to the right shows a grass root hair, and you can actually see some little dots in there. Those little dots are the protoplasts, but you can see right here at the tip, those are bacteria that have just been moved out through a pore, and then they're reforming their walls there, becoming bigger rod forms, bigger rod shapes. So the plant essentially rebuilds these microbes as soon as it puts them out into the soil, makes them soil competent again. So the way this works is we have a model, we call it the cyclosis expansion wave mechanism. But essentially what this is that what the microbes here in this diagram are these little black dots. And essentially what happens in this dotted line is the protoplast of the root cell. And so what's happening here is the what we call cyclosis, and that is the plant is moving these microbes, moving the cytoplasm around like this, constant rotation, moving those microbes and depositing them out at the tip where they're accumulating out here. And that's why this tip is becoming fat, because those microbes are getting stuck there. But every now and then one of those microbes is caught again and it's moved down again, replicated some more and broken and so forth, and then cloned, and you have more of them formed. And then what happens at some point is there is an expansion wave in this hair. And what happens is there is a little, almost like a pressure bubble that starts at the base and propagates, goes to the tip. And as it does, it pushes this protoplast, this membrane gets pushed out. And as it does, it squeezes those microbes out those pores at the tip, and then they go back out into the soil. So it is actually the plant is ejecting those microbes back out into the soil. Okay, and this is another one of our test plants, and you'll see this process, parts of this process in these test plants. This is a sedge, and these things grow. These particular sedges grow on these limestone rocks, so they grow without soil, and they do pretty well without soil. But they do have these microbes, and they are doing a lot of rhizophagy cycling with these microbes. And see, so this actually is, if you look at the bottom one is stained for reactive oxygen. And you can see these dark brown or reddish areas. And you can see this one here, this little one has some red there, and you see the blue there. That's the microbe there. So it's outside that membrane. Here's another one, microbe. And that red area, that's where all the, in this case, hydrogen peroxide, but all the superoxide was concentrated right around there. And these things are in constant motion. They are circulated around with cyclosis, this circular movement, moving them around. We think that's important because what that does is that will actually break down concentration gradients so that nutrients can go very rapidly between the microbe and the plant, or in some cases the other way, because the plant is feeding these microbes with carbohydrate. It is keeping them going with carbohydrate, but many other nutrients are being extracted from them. This top, this is another root hair, but this is a movie actually that shows these microbes. You can see the little bumps here. These are where the microbes are circulating around. They're constantly being circulated around. And of course, that adds to this cloning process by breaking up the microbes and facilitating nutrient exchange. So it's a very active process. You don't usually see root hairs like this moving, but they do this. This is what they do. To get these pictures, what we do is we germinate on agarose on a Petri dish, a clear plastic Petri dish, and then we turn it upside down and look at it in a microscope. And so that's why we can see this movement. This is moving about as the right speed for these, maybe a little bit faster. These things move about 10 micrometers per second. So they actually are moving quite rapidly in these hairs. So this is a still photograph again, but you can see all these clusters of bacteria, of these protoplasts in these cells. And of course, these clusters, we think all of these clusters develop from one or maybe a couple of cells that were jostled apart and broken apart as they were moved around. So this is part of this cloning process that the plant is doing. So this is that sedge root hair, and you can actually see now the whole process. You can see the tip here, you see the movement, you see the cyclosis, those microbes circulating around. You can see them being deposited here at the tip, and these are jostling around. And when we stain it with superoxide, you can see much of the superoxide is concentrated there where those microbes are. So, you know, the growth, how they actually are triggering development, we don't know. We think it may be some type of reactive oxygen signaling that the microbes are producing, nitric oxide or something that's causing elongation to occur. We haven't yet deciphered exactly how that's working. So this shows one of these sedge hairs, and you can see all the microbes. See how fat they are at the tip. These are fat with microbes. You don't see the microbes in there because the only ones that touch the membrane actually show the staining. But there's a lot of microbes in there actually that aren't staining because they're not touching the membrane. But you see these here that are. You can see they're red, and you can see where the microbes were ejected out. Right here, you can see all these here outside the hair. They're reforming their cell walls here outside the tip here. And this process is shown here. And this is actually a tomato. And what you can see is that that there is a pressure that occurs, an expansion wave that occurs in the base of the hair and then propagates to the tip. And see, as it propagates, it forces those microbes out of that tip. And you can see it goes here, you can see all the way from there. Now, what we think is going on is that the root cells are, for some reason, they're triggered to put potassium into the vacuole in these hairs. And that's causing water to rush into the vacuole, and that's causing this expansion wave that happens at the base and then propagates to the tip. That's how it ejects. Now, this happens to be a process that is actually periodic. So it appears to occur, periodically, let's say every few hours, but we don't know what exactly triggers it. We have not figured that out. We do know that we can put more potassium in the medium that these microbes that these plants or seedlings are growing in, and especially with a little bit of nitrogen added, they will then eject microbes faster. But we don't know what it is that triggers the ejection. We know it's periodic ejection. As it turns out, and this is also counterproductive or counterintuitive, counter what we know about hairs, we think that hairs function to absorb nutrients, which would mean the nutrients are actually going outside to inside. But in this case, in terms of the rhizophagy cycle, at least for carbohydrates, it looks like the nutrients are going the other way. That the youngest bacteria, in this case it's a yeast here, the youngest one is seen right there at the tip. The protoplast stages are underneath here. You can see these little spherical structures. All these over here are the protoplast stages. The youngest one's here, but then they grow and get their cell walls back out here. So the plant, in order to do that, the plant has to be sending nutrients out to those microbes out there outside once it ejects those microbes out. Okay, so what happens to plants if we don't have the rhizophagy cycle? So what happens is essentially this. Okay, this is rice. What we did is we removed the microbes from the surface of the rice seeds using that 1 hour sterilization, using also a little bit of antibiotic to make sure we got every little last thing, and then we washed all that off, cleaned them off. And then we put our pseudomonads from Phragmites, put them back on this seed over here, the E+, where we have pseudomonads, and you can see these seedlings are big. And then we left them off of this seed over here. And you can see it's tiny, it's very small, it didn't develop. There's a huge growth inhibition effect when those microbes are not present. This is a little bit later on from the same experiment. Here's with the microbes. And you can see the roots. You can see all the soil adhering to those roots. The microbes change the whole relationship of the plant with the soil. The soil now holds very closely. It's a very tight relationship with the with the plant. Also, there's hairs here, all out here, and the hairs have something to do with holding that soil there as well. But also there's microbial exudates, there's plant exudates, there's everything doing that. So the rhizophagy cycle, the microbes there really make a difference in terms of that plant health and relationship with the soil. Here's for the one with no microbes, where we left them off, E-. And you can see there's almost no soil, very little soil adhering. There's also very few root hairs there. The root hairs were suppressed. So it makes a huge difference in terms of development. So what nutrients? So we've done a series of experiments over these past couple of years to try to figure out what nutrients were. And I'm going to describe one done by a visiting postdoc in the lab using wheat. And what he did essentially was to take the wheat, isolate bacteria off of wheat. So these are the native microbes that came with the wheat seeds. And then we sterilized the wheat seeds, and then he put them into soil, in this case, sterile soil. And then he put some with no microbes, some with the various microbes that he pulled off of the wheat, the endophytic microbes. And what you see here is this. Okay, so these are the wheat seedlings. And this is with no bacteria on it. I say the no bacterial control. And you can see these have little tiny roots. And the shoots are also small, stunted essentially. And to the right, you see this is with one of the microbes put back on. You can see these shoots are big. They're green. The roots are big, well-developed, long. They also have lots of hairs, although you can't see that here. It makes a huge difference. Okay, now then what we did is we took the shoots and then we measured the nutrients in them, dried them and measured the nutrients. And this is what you see. This is without bacteria to the left, number 1 here. And you see the bar comes up this high. And then this is he had 3 different bacteria that came out. They're different bacillus species. Bacillus, actually, these are 2 bacillus species here, the same. And then we have a second bacillus. So there's actually just 2 different bacteria here. But you can see with the bacteria, you see there's probably about a 30% increase in the amount of nitrogen that's going into those shoots. So it's making a big difference in nitrogen. This is for phosphorus. It makes a difference in phosphorus. That may be a 25% increase in terms of phosphorus when you have the microbes there compared to not. With potassium, it looks like with the right microbe, with this particular one, it looks like maybe 3 times the amount that you get without bacteria. So phosphorus is with that bacteria the plants get a lot more phosphorus. What was that? Potassium, a lot more potassium. Calcium also, it looks like about a 25% increase when the microbes are there. Sulfur, you can see an increase. The microbes are important in sulfur acquisition. Manganese, you see the same difference, the same pattern. Magnesium, the same pattern. Zinc, the same pattern. Now, there is some work that's being done now related to climate change. And this is actually with this work, what people are doing is they're putting into the field, they're putting carbon dioxide, they're kind of squirting carbon dioxide in these little enclosures so that carbon dioxide will go out into whatever the crop is. And then they will measure nutrients in those crops, in the crops that are harvested. And what they're finding is that in a lot of C3 plants, there's declines in terms of protein, magnesium, iron, and a lot of other nutrients. Many nutrients are affected. Soybeans, they have rhizobia, so they're getting the nitrogen, but they show all these other nutrients. Corn and sorghum, they're less affected by the carbon dioxide, but corn and sorghum, they're C4 plants, and they kind of handle carbon dioxide a little bit differently than the C3 plants. So they may be sequestering carbon dioxide, so it may not be having a negative effect. What we think is happening here is that the carbon dioxide is actually going down into the soil. And because it's heavier than air, it'll accumulate in the soil. And so carbon dioxide is going, what we think is happening, carbon dioxide is going very high in the soil. And carbon dioxide actually will inhibit the rhizophagy cycle. And what the CO2 does, carbon dioxide, is it will inhibit superoxide production. So if it's high in the soil, high carbon dioxide in the soil, then it will suppress actually rhizophagy cycle and extraction of nutrients from microbes. And that will also suppress the capacity of the plants to clone these microbes and to increase them and put them into the soil. So it has negative effects on the rhizophagy cycle and on nutrients in plants. Now, as it turns out, the levels that we use in experiments and that are probably going into the soil in these experiments are much higher than people use in greenhouses, where they are actually putting carbon dioxide into the air. So we use much higher levels, and probably in the soils and also much higher levels accumulating in those carbon dioxide enclosure experiments. Okay, but we do experiments by using elevated CO2, and we use them. What we'll do is we'll grow plants in these little containers without the lid. This has a lid on it, but without the lid, and we'll put them in these chambers. And in these chambers, we will put high levels of carbon dioxide, and then grow the plants for a period, and then analyze what the effect it has on the plants. Okay, and so this shows tall fescue seedling, and you can see the one to the left. This is in regular air. You can see all the superoxide, the purple there, all around the tip where those microbes are being hit with superoxide, and you can see it has long hairs there. And this, the one to the right, was grown in elevated carbon dioxide, and you can see there's not a lot of blue there. There actually is in the center some blue, but all the superoxide is being suppressed around the surface, and the surface is where the microbes are. That's where they're being processed. So you don't see any there. And also notice you don't see the hairs, not like you see over here. Hairs are inhibited. Hair formation is inhibited because those microbes aren't replicated in the root cells. You don't get hair formation, or it's very spotty. Like here it is. This is an older part. You can see all the hairs. Here's one that's been in elevated carbon dioxide. You can see the hairs are short. This is stained also for superoxide. You see no superoxide out there because there's no microbes out there, or the microbes are not replicating like they should, so you don't get good hair development. Okay, so we did a similar experiment with wheat using elevated carbon dioxide, and this actually shows a result of that. And here, this actually is the root hair in elevated carbon dioxide. And you can see these are long needle-like root hairs. They're not fat. And these are stained. You should see microbes here. There's no microbes in there. You can't see any microbes. These happen to be, now we can actually get hair formation if we grow seedlings on agarose that contains some protein. So protein will trigger hair formation. And we don't know what about protein does it, but it could be that protein from the microbes is also what triggers hair formation. We don't actually know. Okay, but look to the right now. This is in regular air, and you can see these are actually fat. These are the tips these are the same magnification, and you can see the microbes in there, the little dots in there. Those are all the microbes inside that hair. Here, the bottom one, you can see it's real fat there, and you can see the blue there. That's all where the microbes are in there. Okay, so that's in regular air, but you elevate it and no microbes form, and you can then see the true shape of the root hair actually is this needle-like structure rather than this club-like structure. Okay, this is a close-up now of the high elevated carbon dioxide, and you can see these hairs again. There's no microbes out there, but there is. And you can see here, you can see where the arrows are, these little blue things, those are rods. What happened is because we inhibited superoxide, those bacteria never lost their cell walls. they kept their cell walls and they kept their rod shapes. And they're also not, they're in these little clusters now in there, and they're not circulating. So they're not being replicated. At least that's apparently that's how we interpret what we're seeing here. We've seen it in many experiments, similar effect, the similar thing. The CO2 is inhibiting the rhizophagy cycle by inhibiting superoxide formation. Okay, so this is winter wheat. Okay, we did a similar experiment in soil, and this is regular air. And you can see the roots with all the soil adhering. And this is an elevated carbon dioxide. You see there's very little soil adhering to that. The CO2 is inhibiting the rhizophagy cycle. So we're not getting that proper development and soil absorption. We're not getting nutrients being carried back to the plant. At least in that experiment, that's what it appears to be. Okay, so this is a close-up of a root formed from that experiment. This is the wheat. You can see this is regular air. You can see all the hairs there. This is normal. This is to the right is an elevated carbon dioxide. And you see no hairs or very scant hairs forming there. It makes a big difference. Without the rhizophagy cycle, you don't get those microbes forming. You don't get the proper development. So I have a student who's studying this, and he's using rice. And what he did was to take the rice, grow it in regular air, and grow it in elevated carbon dioxide environment, and also rice with the microbes totally removed. And so in this work, okay, what he showed, this is just actually measures, this is from that experiment. This is the carbon dioxide on rice. And he shows actually to the treatment over to the left, all the way to the left, no microbes in air, no microbes. Okay, the microbes are removed. And this is regular air. So these plants, this is actually the length of shoots and roots here and shoots here. You can see these plants are pretty small. This is just, again, the length. And this is no microbes plus CO2. You can see the plants are small. It's inhibiting development. Okay, when you remove those microbes, it completely inhibits development. Okay, now, and this treatment, okay, this is with microbes in air, regular air, and you can see the plants are like this. You have the shoots may be a similar size. The roots are pretty big, okay? And this is in the elevated, the one all the way to the right, this is in the elevated carbon dioxide. And this actually shows you that CO2 fertilization effect. You can see these shoots are bigger, and you can see the roots are a little bit smaller, or actually those are definitely not different. So the plants appear a little bit different, a little bit bigger though. So you see that CO2 fertilization. We know that happens, but it's the nutrient effect, that the reduction in nutrients that people aren't clear on. Okay, and so what he did then was he took the shoots and the roots, and then he measured the nutrients in them. And this actually shows phosphorus. And this is this big table here. This one microbes, the one microbes plus air. These are with microbes and regular air where you get maximum benefit. And you see all this phosphorus. It's a lot of phosphorus in there, double the phosphorus really in this case when you add the roots, shoots and roots together. And if you look at this one, no microbes in regular air, just remove all the microbes, you can see it's less than half of the phosphorus that these plants are getting without those microbes. Okay, but then here's with the elevated CO2. Okay, so that suppresses superoxide, that suppresses nutrient extraction from the microbes. So he put microbes here, but he increased the CO2, and you see what happened is the phosphorus is half what you get in regular air. So rhizophagy is delivering phosphorus. That plants appear to be getting this phosphorus from these microbes through direct oxidative extraction. The same is for zinc. Okay, zinc If you look at this, microbes in the air, all this zinc. This is no microbes. Plus, there may be some microbes that are there, and they're probably getting some. But this is in the elevated CO2, and it's like less than half of the zinc. So these nutrients, phosphorus and zinc, are being extracted oxidatively from the microbes. Okay, so this is sulfur, and you see for sulfur, you can see actually without the microbes, you got a very little, but with CO2, you actually see even a little bit more sulfur. This is a nutrient that the microbes are delivering to the plant by solubilizing in the soil, and then the plant will absorb that nutrient from the soil solution. And the CO2 actually increases it, probably because we have carbonic acid, and that's forming in the soil water, and that's leaching some of the sulfur out that the plants can then absorb. Okay, so this is potassium. You can see it's a very similar thing. You can see here, you put CO2, potassium is about the same as in regular air, but you remove the microbes and it reduces it. So this is also solubilized. Potassium is solubilized in the soil. That's what we think. That's how we're interpreting this. Manganese, same thing. It is solubilized in the soil by the microbes, and then the plant absorbs it from the soil solution. Boron, same thing, same deal. Microbes will solubilize it. Plants can get it. But if you remove the microbes, they get less. So even for boron. So you have these different nutrients. It turns out that rhizophagy cycle is delivering this through oxidative extraction. It's delivering the same nutrients that mycorrhizae deliver. They deliver phosphorus, they deliver zinc, and they deliver nitrogen. Except probably here, rhizophagy cycle is delivering more nitrogen than you have coming from mycorrhizae. Although we're still testing to see exactly how much. So a lot of our seeds have lost their microbes because of the way we treat seeds. And what they do with cotton is an example. What they do with cotton is, in order to plant cotton, they will remove all the cotton fibers, all the hairs, the cotton fibers on the seeds. And to do that, they treat it with acid, and to get these seeds nice and clean. Well, as it turns out, the acid will actually kill the microbes on the cotton. And the microbes are carried on those fibers on the cotton, and also on the surface of the plant, of the surface of the seed. You can see here some fibers. You can see these little blue things in here. Those are all the microbes in those cotton fibers. So it's the vehicle where cotton gets those microbes. There you see the bacteria on them, but also on the surface. This actually is the surface of the seed coat, the cotton, and you can see the little dots there. All those little dots, okay, those are the bacteria there. That's the surface of the seed coat of cotton. And so what happens is cotton has the reputation of being a really dirty plant. And we think that's because we've lost the beneficial microbes that carried on those cotton seeds in our planting. And what really compounds this is what we've done to the soils, with soils that where we've damaged the microbial community. And so the plants then can't get good microbes out of the soil. And so what people have to do with cotton, they've got to squirt lots of nitrogen on it, lots of fertilizers on it, lots of insecticides and fungicides and so forth in order to cultivate it. So it has a reputation of being a dirty crop because of that. What my student did is she took microbes from wild cotton and cotton relatives, and then she put that on cotton seeds that had been treated with acid to remove the fibers, and that removes the microbes. And what she found is this. Okay, when she this is actually seedlings, this is cotton seedlings, and this is without the microbes, just grown in soil without the microbes to the right, without the bacteria. And this is with one of the bacteria put back on. And you can see the huge difference. this is the bacterium from wild cotton that was put back on cotton. So we're treating seeds in a lot of different ways, but a lot of what we do to seeds actually reduces the community of microbes on those seeds. So we are damaging the natural community that seedling, that nature intended for that seedling to have when it germinated. But these microbes also have another effect. They will change the chemical composition of plants. And we did an example here is one experiment that we did using carrot seedlings. And what we did was to take bacteria off of carrot family, different off of celery, off of parsley, off of cumin, took bacteria off of these carrot relatives, and then we put them on carrots to see what it did to the chemical composition. And here you actually can see the root hairs of the carrot, and you can see the bacteria, these little dots in here, in those root hairs. And so we know the rhizophagy cycle, the same thing happens here that happens in all other plants. This is actually the bacteria in the root epidermal cells. You can see they're in clusters, all these brown clusters of things. Those are all the bacteria. This is stained with that DAB stain that we use. And this shows the alpha keratines. This is now analyzed, the root tissue was analyzed, and you can see without bacteria, the control there. Here's one of the bacteria that were put on. It didn't have any effect. Here's another bacterium 2. It doubled the alpha keratines, and bacterium 3 increased it a little bit. So different microbes are going to have different impacts on the chemical composition of plants. And of course, this can affect the healthful compounds that plants naturally have. This is the beta carotenes, and you see the same thing happening with beta carotenes. Just bacterium 2 markedly increased the beta carotenes forming in the carrot. Can we repair our crops where we've damaged them? Seeds, for example, where we damaged them by removing microbes. And so I have a student who's working on that, and she actually did an experiment using some commercial biostimulant microbes to see if they work. Her work was done in celery, and what she did basically was to sterilize the celery and then take these biostimulant microbes, put them onto the seeds and let them germinate and evaluate what happened. Now, it turns out not all biostimulant microbes are equal. And most of these biostimulants have mixtures of microbes. And so some are better than others. Some of the microbes work good and some of them don't, depending on what plant you have. This actually is the control, no microbes. You can see the celery has no root hairs here. Here you can see with one of the biostimulant Bacillus amyloliquefaciens, different species work too. It doesn't have to be just this one species. It doesn't have to just be Bacillus. It could be other species of bacteria. And you see the hairs are forming there. Okay, so we can, and some of these biostimulants work. Okay, so you can see here, this is with this commercial strain again, and you can see the root hair, and you can see all the bacteria in it. See the blue here, that's bacteria outside. And here you can see hair, and you can see the bacteria inside, and you can see some of the bacteria outside. And there you see bacteria inside those hairs. You can see some of the bacteria outside. Actually, you see right here, they're actually coming out. And when they come out here, they're forming chains for this particular bacterium outside the tip. This is with a different kind of stain. So we're exploring these different ways to visualize these microbes. This is iron stain. And this is a cyanoferric stain, or it's a stain that it's actually a stain for iron, and it will stain blue if there's iron, and these microbes are retaining iron, and so you can actually see the iron, the blue in the microbe, because they're holding on to iron. And if you measure, actually you can measure the plant. If you grow grasses without microbes, you'll see that the shoots actually have more iron inside the shoot. They have very high levels of iron. In fact, they may, in some cases, might even be toxic levels. But with the microbes, it pulls it down a little bit, because those microbes themselves are holding on to some of that iron. That's important, because the microbes need iron too. And one thing they need iron for is nitrogen fixation from the air. So that makes us think that they may be fixing some nitrogen actually in the root hairs. We're at least checking that. That's not a guarantee, but that's one thing we're looking at. That shows it again, commercial Bacillus strain. There it is. There you can see the red arrows. You can see them coming out. There you see, again, that iron stain. You can see them in there, and you can see them coming out again. So these, in the rhizophagy cycle, plants get nutrients from the soil, from that microbial community in the soil. Those nutrients flow to the plant. The nutrients that the plant needs. The plant has abundant carbohydrate, so it secretes out of the roots these carbohydrate and some other things that it has in abundance to feed that microbial community, keep it going. And in the process, the plant becomes, for one thing, the plant gets nutrients. Up here, number one, absorbs those nutrients, so the plants grow better. The plants become more oxidatively stress tolerant. And that's partially because they're using reactive oxygen to get nutrients out of the microbes. And the other benefit is that these endophytes go into the soil, these bacteria for the most part, and then they colonize fungal pathogens and make them avirulent. They don't necessarily kill them. Instead, they colonize them and they cause porosity in their membranes, and they get the nutrients that leak out of those pathogenic fungi. But the outcome is those fungi cannot cause disease as a result, because they don't have enough energy to do that. So some points that I want to make is that seeds should be carefully managed to maintain the native microbes. So they should be treated in such a way to preserve those microbes. And that soils should be managed just as the seed should be managed to build up the microbial communities in the soils. Okay, so that's very much regenerative agriculture. And third, that biostimulant microbes are one way that we can essentially heal the damage we've done to seeds, provided those biostimulant microbes actually function in the rhizophagy cycle and go into the root, and many of them do. It's not to say that all of them do, but many of them do. Those are some references, some articles done over the past couple of years. If you want to learn something about, go deeper into this, you can look into those references. Okay, these are all the people who contributed to this work over the years. It's been a lot of people. Some people who aren't even listed here have made contributions. And that is it. And that's it.
Well, Dr. White, I don't know where to even start with questions. Remarkable research, basically showing, how the biology interacts with the plant, and what you showed there about nutrient density was absolutely amazing, and the nutrients that the biology is providing back to the plant. So I know I read in some of your research, you were talking about like nitrogen efficiency when the rhizophagy process was occurring. Could you expand on that just a little bit?
Yes. So what we see is that when the plants have their rhizophagy microbes, they're getting about 30% more nitrogen than when those microbes are absent. And we think that much of that nitrogen is coming directly off the surfaces of the bacteria, because its cell walls are stripped off and they have their organic forms of nitrogen there that the plant can absorb. There may be some, and what we also know that when the microbes are there, the plants will upregulate nitrate transporters in the roots, in the root cells. So they're actually, probably what's happening is they are oxidizing those organic forms of nitrogen all the way down to nitrate, or certainly there's some nitrate there. They also may be absorbing some of the organic forms directly. We don't know all the things that are happening. We also think that, at least we hypothesize, that there may be some hidden nitrogen fixation that's happening in the roots, in these root hairs. And the reason we say that, the reason we hypothesize that, and it seems counterintuitive because there's reactive oxygen there, but what's happening is that by subjecting these microbes to reactive superoxide, what the plant is doing is, besides making it leak nutrients, they're making these microbes upregulate their antioxidants and other oxidative superoxide and other reactive oxygen scavengers. So they may internally be very low in reactive oxygen. And so the other thing is that the plant is replicating these microbes. And so some of the protein that they brought in, because they're replicated, they're essentially losing. So what the plant could be doing is attenuating those nitrogen reserves inside the microbes that it internalizes, forcing some of those microbes to actually fix nitrogen. We don't know exactly how it's happening. The other issue is, and I pointed it out, these microbes are holding onto iron, and iron is one of the cofactors, one of the nutrients that these microbes need to fix iron, to fix nitrogen. In the iron, there's an iron protein actually involved in the nitrogenase. So for a lot of reasons, we think that rhizophagy cycle is important for nitrogen acquisition.
Absolutely. We got a question here. What magnification are you looking at the root hairs?
Those would be, most of those are actually, most of those are under 1,000, 100 times objective lens with a 10 times ocular. So it's about 1,000 times magnification, something like that. So they're pretty highly magnified.
Another question here. Is the plant selective in the microbes it cultivates?
It is. The plant selects the microbes it cultivates and internalizes, and probably it's internal. We don't know the complexity of this process, but the plant's not taking in one microbe at a time. It's taking in multiple microbes from the environment, and some of those from the soil, and some of those microbes are likely fully degraded by the plant, and others are not. And the ones that are not fully degraded, the ones that survive that process, are the ones that trigger root hair elongation, and they're the ones that are replicated in the root hairs and then shot back out into the soil. So those are the ones that match the plant, the strains that the plant is using most efficiently, we think.
Thank you. I know you addressed this, but just to reiterate this, there's a question saying, are the microbes on dormant seed?
They are on dormant seed. The microbes actually will, they will go dormant themselves. And some of them are, like a lot of grasses will have a lot of bacillus. These are endospore formers, so they're able to go dormant, and they're very, very resistant on the seed. But then we have others that don't form endospores, and they're nevertheless in that seed. Now, how they're surviving on the seed, we think they form biofilms, and somehow they're becoming resistant in those biofilms. We don't exactly know the full process of how they're surviving. We do know that you have to actually, a seed has to develop properly to have its microbes community develop properly, and that generally we've done work with grass seeds produced in very dry environments where they get little moisture, and then those done in wet environments where they get a lot of moisture, or an average amount of moisture. The ones in dry environments don't develop good microbial community on the seeds, and when those seeds are planted, what happens is soil pathogens get them and kill a lot of them with damping off disease, for example. And so you don't get good grass stands. But in moist environments, you get a high diversity of microbes and lots of cells on those lots of microbial cells on the seeds, and then those develop properly. They have little disease, and you get better stands in turf grasses.
I mean, that's a great observation and great information, definitely for selecting seed, especially a lot of our native seeds. It's very expensive to plant, where you're sourcing your seed from. Another question here, can superoxides break down virus coatings as well?
Yes, the superoxide is highly potent. It would zap anything. Yeah. So, I mean, in fact, I should go into that a little further. Humans, any eukaryotes, plants will use this same system to attack pathogens, for example, and this is part of our innate immune system that our cells and plant cells will use superoxide if it detects, say, a pathogen, for example, a virus that goes on into the tissues, detects it, and then it recognize it as foreign, something different, and then it will shoot superoxide onto it in an attempt to degrade it.
Thank you. Another question here is, what effect does synthetic fertilizer have on rhizophagy process?
We don't know all the effects, but we do know that it suppresses plants doing rhizophagy. It tends to suppress the rhizophagy cycle. What happens is when their plants like corn, for example, are growing without nitrogen fertilizer and using their microbes instead, what will happen is they will make shallow roots with lots of branches and lots of root hairs. So you'll have lots and a certain layer of the soil. But when you put inorganic nitrogen, for example, squirt fertilizer in it, they don't grow like that. Instead, the roots will be long and grow longer down into the soil. And they won't do rhizophagy cycle. And of course, if there's anything like nitrogen fixation occurring with the rhizophagy cycle in the roots, then the roots would need to be at a certain soil level where there's a certain amount of oxygen. And so the roots wouldn't grow deep like that. So from observations like that, we think that putting inorganic fertilizers, certainly too much inorganic fertilizer, or over-relying on inorganic fertilizer would impact negatively on the rhizophagy cycle.
The next question is, does rhizophagy occur in legumes and brassicas?
Yes, it occurs in legumes. You saw the images from clover. Clover does it. We've also used certain commercial biostimulant microbes on clover as well, and they also go in, and you see the rhizophagy cycle occurring there. This is a different process. This rhizophagy cycle, all or most plants can do rhizophagy cycle, at least at some stage in their development. Even legumes. Now, as it turns out, legumes will develop the nodules, as you know, and nitrogen fixation will happen in those nodules, so they'll get extra nitrogen. Okay, so that's why legumes appear not to be impacted as much by this carbon dioxide that people are doing in these experiments, and they find that nitrogen supply is not being affected if they have CO2 there. If they suppress the rhizophagy cycle using CO2, in legumes, it does not impact on the nitrogen because the legumes get their nitrogen from those nodules, from rhizobia.
Absolutely. When you were doing the experimentation in regards to CO2, it made me think, when our soil's crust, definitely after tillage, our management practice such as that, we seal that soil surface off, which will, increase the CO2 levels within the soil. So, showing how our management can, really dramatically have some negative consequences in nutrient cycling due to this process.
Yes, and in fact, it could be that what's happening in a lot of those soils is we're getting too much CO2, high levels of CO2, and that's suppressing rhizophagy. It makes all the sense in the world. And it could also be a solution. I mean, there could be a way that we could say, for example, in aerating soils, one might be able to say inject oxygen into soils that have too much CO2 and reverse the inhibition. I don't know. We haven't gotten that far, but there may be some ways to counteract that elevated CO2 rhizophagy suppression.
What modern seed handling or coating techniques reduce viability of bacteria on seed?
Yeah, so that's a good question. For a lot of seed, okay, like for example, the acid treatment of cotton, that does it. Another treatment is the hulling of seeds. Some seeds, for example, grass seeds like Bermuda grass, those seeds are hulled. And as it turns out, the microbes are actually carried on those hulls. And so hulling them reduces the microbes that are there on the seed. And another thing that we do, sometimes we'll put, then we have these hulled seeds, and then we soak them in fungicides, which also has impact on those microbes.
Okay.
Yeah, so any kind of seed treatment, any kind of sterilization of seeds, removing material on the surface of the seeds, too much, even too much cleanliness, seed hygienics. People think, well, you need to take care of these seeds. Don't let them touch the ground. Don't let them sit out there. Don't let them get rained on. All of that is impacting the microbial, because plants actually are evolved to have those seeds out there maturing with moisture around while those microbes the microbial community is developing on the surface of those, at least those kind of like grass seeds, for example.
Yeah, I think another great question here. We're just I think you've touched on this, but would the nutrient absorption process in the control with no root tip microbes be different from that with root tip microbes?
Yes, it makes a big difference. If you take away the microbes, the nutrients is cut drastically for most nutrients. Ironically, iron is the exception, which strangely, we had in that one, what we actually see if we in wheat, in our wheat experiment, if we take away the microbes from the wheat roots, we don't give them any microbes, then we see the wheat getting very high levels of iron in the shoot. You put the microbes there, and then more of that iron stays in the root with those microbes. So it reshifts, it moves where those nutrients are. And so iron is the curious, most interesting exception to that rule.
Interesting. We had another great question. Does fire, we have a lot of, especially in my part of the world, warm season grasses. We burn a lot of rangeland, where a lot of plants may have went to seed. And does the burning inhibit rhizophagy cycle, or does it inhibit, I guess, more the microbes or damage them?
You know what? Yes, if the soil gets hot, but I don't know how hot it gets down in the soil. I don't know very much about the burning of lands. I know it releases some nutrients, and so you oftentimes will see an increase in plant growth where you burn. But if it's getting hot down in the soil, then that's a sterilization effect that could be happening. If it gets very hot, then you're destroying the whole microbial community.
I've got a question here. They're asking what you're calling hairs. Is it really mycorrhizal?
No, they actually are root hairs. Well, I didn't show any images of mycorrhizae in this case. They are all root hairs. They're all plant cells. They're the extensions of the root epidermis that go out, and that we've always learned to go out to absorb nutrients out into the soil. So they are actually root cells. They're the root cells themselves. So those microbes are going inside the root cells. They're not mycorrhizae. I didn't show any mycorrhizae. It just turns out that the nutrients that plants get from mycorrhizae are the same nutrients that plants are getting through direct oxidation extraction from the microbes. The microbes give all kinds of other nutrients by solubilizing it, and then the plant going to the soil, solubilizing it, and then the plant can absorb it. But nitrogen, phosphorus, and zinc are the three nutrients that mycorrhizae also provide to plants. So this is another symbiosis in which the plant essentially is farming these microbes to extract nutrients from them.
Thank you. You know, one thing I noticed when you showed that one illustration, you know, the roots with the rhizophagy cycle occurring, you know, those young roots were already starting to develop soil aggregates along that rhizosphere, along that root. And that's one thing we talk a lot about, you know, getting our soils aggregated. And, you know, it's amazing how the plant's, you know, communicating with the biology to get that started itself. And when we inhibit that by not having the microbes, you know, we really deter, I mean, from building in the soil aggregate. I mean,
Yeah, it's really amazing. It's really amazing. We think that is a combination of when the microbes are there, the plants are feeding them with carbohydrate exudates, right? And those go out and they will feed those microbes out there, but also the microbes growing in those exudates will bind that soil particles together. And so that the plant essentially has basically the microbe is the conduit, conduit, right? The means by which the microbes are the means by which the plant are getting those soil particles, that nutrients from those soil particles.
We got a great comment here from one of the attendees, you know, that we should be inoculating seeds with excellent biology and nothing else.
Well, that's what I think.
Well, it obviously kind of looks that way, doesn't it?
Yeah, that's what I think. With the right biology and nothing else. Yeah, that's what I think. I actually think regenerative agriculture really has it right. Someday we're going to be, most farmers are going to be farming using microbes more than chemicals. Of course, you know, there's still probably going to be fertilizer applications of some sort. You know, soils that have no fertilizers and no kind of nutrients in them will have to be, you know, you have to put something there. But I think that with the microbes and with the organic community, develop the organic community, the plant community, there's going to be, you know, people are going to be doing that more and more. And that's going to have benefits for the environment because a lot less runoff of fertilizer, a lot less damage to the environment by doing that.
Oh, absolutely. That's what regenerative agriculture is all about. Another question here. Are these bacteria you have worked with native to non-cultivated soil or composted organic materials?
No, they're quite native in both organic materials and native soils. They're quite a normal part of the soil community.
Do you have time for a few more questions here this evening? And
sure.
Wrap it up. I know you've given us a lot of your valuable time. Would genetically modified seeds that have been selected under high synthetic fertilizer use have a lower ability for rhizophagy?
Yes, they would actually. What happens is what happens is when we produce that material in the laboratory, typically we're using tissue cultures, and tissue cultured material is sterilized, or many of the microbes are removed, and so the plants produced from that sterilized material then lack the native microbes that would normally be there. And, particularly, if those, so right away the seeds are compromised because they don't have their beneficial microbes. And what often will happen is that what that does to the plant is it will make it totally dependent on inorganic fertilizers for nutrients without those microbes. Now, I mean, if there is a healthy soil that you could plant these seeds in, the plants can reacquire many of these microbes from that soil because that's where they come from. These plants fish these microbes out of the soil. They select them from the soil, and then they keep the beneficial ones and then put them on the seeds so that the seeds will carry that beneficial microbes with them.
You made a comment early on about, plants that don't have, the beneficial bacteria on them or in them, how that affects root development and growth. And one observation I've made several times throughout the years, especially on corn, is we often see corn root systems very shallow, laterally growing, and not really developing much of a taproot. And we've often attributed that to, hard pans inhibiting, the roots to go deeper. But, after reading and hearing a lot of your research, I'm starting to wonder if we haven't inhibited this rhizophagy cycle to the extent that that's what's causing this more, possibly.
That could be.
And things. So we got about 2 more questions here. We'll wrap it up this evening. How can we manage seed collection to preserve the bacteria?
That's a good question. Actually, if the seeds are produced under conditions, say with moisture, in which the microbes are able to develop, with there's moisture out there, for example, then the microbes will survive on those seeds for a good period of time. We've had them actually surviving on turfgrass seeds for years on the surface, and we store those turfgrass seeds in a refrigerator, nothing more than a refrigerator, and they've lasted. The microbes are still there after 5, 6, 7, 8 years. So the microbes on those seeds are surprisingly resistant. But if you remove those coatings or you put antimicrobials on the seed or treat them with acid to clean them or something like that, then you damage the microbial community. So, I mean, if you grow them properly, the microbes will be there. Another example, actually, is there was an experiment that some investigators did using a wild strain of tobacco. And what they did is they took this wild tobacco and they put it in cultivation out west. I think it was done in Utah, I think. And then they would harvest every year their seeds and they would take it in and then they would store it. And then the next spring they would plant again. And then they did this for about 7 years. This cycling, going, harvesting, going, storing it, and then planting it again. And what happened after 7 years is they started seeing a disease, a bad disease that was wiping out the tobacco. And then they went back and they started looking at their original plants, and they isolated some microbes from that plant. And then they took those microbes and then they put them back on the seeds, and then they cured their tobacco seeds from that. Was actually a wilt disease that happened. So they cured it from that wilt disease by re-putting those microbes back on that native tobacco. So what's happened is they would gather the seed that normally would stay out there in the field all winter long. That's what happens in nature. But because they would gather it and take it in and put it in a dry place, the microbial community was little by little being lost and being altered. And so then those beneficial microbes weren't there after 7 years to protect those little wild tobacco plants from pathogens, from damping off pathogens or wilt pathogens.
Amazing stuff. This is a great question. Are microbes biostimulants, or do microbes produce biostimulants, hence quorum sensing?
You know, I understand. I don't think quorum sensing actually is happening here. What seems to be happening, because these microbes are internalized, they're not able to secrete very much. They're not able to, I mean, we don't exactly know, but we think they're not able to secrete much because of all the superoxide, because it's zapping any proteins that they may have on the surface that they're trying to secrete, or anything that they're secreting might get zapped. Now, we don't know the details. We don't exactly know. There's a lot of unknown here. So I'm just kind of hypothesizing now. I think not. I think they're not producing biostimulants. I think they themselves are stimulating development by their presence. Maybe it's protein that causes the root hairs to form. For certain, it's probably protein that's causing the superoxide, the plant to detect them and shoot the superoxide on them. It's probably protein on their cell walls that is the trigger for that. And they probably are, these microbes that are trapped inside the cells, they're probably trying to recover and produce more cell wall internally. And then that just gives the plant a more of a capacity to then zap it again with superoxide. And I mean, these microbes, certainly they produce metabolites, secondary metabolites, and they produce auxins, and they produce ethylene, and in some cases gibberellins. They're all growth regulators. And I mean, that all could be happening, but we think it might not be happening because the microbes that are actually active in the rhizophagy cycle, they don't all produce auxin, for example. So we think that it's not really. And in terms of quorum sensing, I mean, at least not the way that I understand quorum sensing. We don't think that's happening because the microbes are really under almost total control by the plant.
No, it's fascinating stuff. So, well, you know, we want to thank you for your time, your research, and sharing it with us. I do want to reach out to our Director of Operations, Kathy Richburg, for helping organize this meeting for this evening. I want to thank again the Soil Health Academy for sponsoring tonight's webinar, and everyone who assisted with the webinar this evening also. Dr. White, we greatly appreciate everything you've done. We look forward to hopefully having you come back and revisiting this in regards to the rhizophagy cycle and you know, how we can, you know, enhance our farming operations by understanding this better. So thank you very much for sharing this evening with us.
You're very welcome. It was my pleasure.
And thanks. So everyone who attended this evening, we thank you for being a participant. Check out Understanding Ag's website. We've got more webinars coming up next week with Dr. Williams, with Gabe Brown, and Ray Archuleta. So be sure to check it out, and we hope to have you attend here soon. So thank you again, and thank you again, Dr. White.
Thank you. Y'all have a good night.
You do the same. Thank you.